Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society. And I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. The views and opinions expressed by our guests are their own and do not necessarily reflect the views of the host, the American Phytopathological Society, or any affiliated entity. This is the 12th and final episode for 2025, and today I'm excited to welcome Dr. Tiffany Lowe-Power, Associate Professor of Plant Pathology at the University of California, Davis. Dr. Lowe-Power holds a BS in biology from Georgia Tech and a PhD in microbiology from the University of Wisconsin-Madison. Dr. Lowe-Power was a USDA NIFA postdoctoral fellow in plant bacteriology at UC Berkeley before joining the faculty of UC Davis in 2019 as an assistant professor. She was promoted to associate professor earlier this year. Dr. Lowe-Power is among one of the world's experts on Ralstonia, a genus of phytopathogenic bacteria that cause lethal wilt diseases of solanaceous crops, ginger, olive, rose, and other plants. She has published almost 36 peer-reviewed papers on Ralstonia and mentored or advised more than 24 grad students, postdocs, and visiting scholars, and more than 75 undergrads. Dr. Lowe-Power is a 2024 recipient of an NSF CAREER Award, investigating the role of toxins in the ecological and evolutionary success of Ralstonia. The award includes a cohort-based undergraduate research experience named Ignite for integrating the next generation of novel investigations of toxin, ecology, and evolution. She currently teaches courses in biology of plant pathogens and population genomics, cohort-based undergraduate research experience. She previously taught a Being a Scientist seminar. Dr. Lowe-Power is an active member of APS, serving as a committee member for both the Diversity and Bacteriology Committee. She is a 2024 recipient of APS's Schroth Faces of the Future Award, as well as a 2021 recipient of APS's Lafayette Frederick Diversity and Mentoring Award. Welcome, Dr. Lowe-Power. It's great to have you here on Plantopia.
Yeah, it's great to be here.
In one of the presentations you shared with me, you called yourself an accidental plant pathologist, and I laughed when I read that. Could you explain that a little bit more?
So I consider myself an accidental plant pathologist, and as I've gone through my career as a plant pathologist, I don't necessarily see that as unique. I think a lot of us come to this field from extremely different backgrounds. I think there's occasionally someone who, really early on knew about the field and came into it, but I think a lot of us are accidental in our own ways. But for my own journey, I knew early on I really liked microbes because in high school, I was lucky to have an amazing biology teacher. Sam Parks had a master's in microbiology. And so she incorporated a lot of that into the freshman bio class I had with her. Mm-hmm. And it was Agnet STEM high school. So there were also a lot of extra STEM classes we could take. Yeah. And Sam taught an actual microbiology course that I think I took as a sophomore in high school. We did Koch's postulates with penicillium on oranges. Through that Koch's postulates, even though that was actually pure plant pathology Koch's postulates, we were really only thinking about it from the original biomedical concept of Koch's postulates with anthrax and cows. It was like, that's unfeasible for the high school classrooms. You did orange, and here's this mold that you see in your kitchen. And so even thinking of the penicillium as a post-harvest was not a part of that.
And when you're talking about high school, is this in Georgia?
Yeah, so Georgia, Cobb County.
Because I know that you went to Georgia Tech for undergrad, and that's in Atlanta, right?
Yep.
So did you grow up in the greater Atlanta area, or is it the county where you grew up far removed from that?
Yeah, Atlanta suburbs. So we're OTP, outside the perimeter.
Oh, okay.
So outside of the perimeter interstate, highway that goes around Atlanta. So yeah, outside, it's an interesting suburb, typical upper middle class suburb that grew up in Atlanta after the 1980s. There's just a lot of development. So I saw an atlas at some point of my town, and it went from 2,000 people to a massive metro. My high school was 2,000 when I was there.
Okay, so you have this really inspiring biology teacher who's incorporating these kind of clinical microbiology methods and kind of experiments. But it didn't just stop there. Obviously, high school was a stepping stone to get you to Georgia Tech. And tell me a little bit about your experience there studying biology and how that kind of pushed you forward.
Yeah, my experience at Georgia Tech actually started in high school. So that same high school bio teacher, she wound up leaving teaching high school and going to get her PhD so she could teach at the college level. She's currently at Georgia State University. And as part of my STEM high school, we did internships as part of our senior year. She had already started her PhD at Georgia Tech, and she was able to network with one of the environmental microbiologist labs to allow me to come in and proceed to mess up a lot of things and not get done for them.
Hey, you've said this in your own presentations, and that making mistakes early on is a really good learning experience. And if you're allowed to make mistakes and kind of guided to make small mistakes, then you won't make those big mistakes later on because you had your hand held during those formative years.
For sure. And I think throughout my career, the failing upwards mantra is pretty key.
Yeah.
It's something I still have in my lab. And there was a really inspiring failing upwards concept I had in my first year of my PhD. So one of the labs I rotated in had what they called the wall of failure, which was like this like pillar, one of the columns that like support the building. And it was this like small, narrow part of the wall that they had all just posted like dramatic failures. There was like a page gel that was just like completely melted and distorted.
Yeah.
Experimental failures. And that was the first rotation I did during my PhD. And just seeing the failures celebrated by these people who were also very skilled. They had papers, they were doing great work, but just seeing their failure in this physical manifestation was just always really useful and like kind of something I would reflect back on when I had my own.
It's really funny because that reminded me of a wall of shame that like my grad students had in the grad student offices, which was a bunch of cubicles for a number of grad students in the division. And they had the same thing, a picture of a gel, or I remember distinctly one phylogenetic tree where they didn't have an outgroup and everything was genetically identical. So it was just a single line with all the names of the isolates. And they're like, that's not really that interesting. But they hung it up on this wall of shame, wall of fame. And I think it's a great idea because the reality is we all have so many failed attempts and so many failures, and we learn from them. And if we don't hang them up in front of us, sometimes we forget that they existed.
Yeah, definitely. It's helpful to move forward with it.
Yeah. So one of the mentors you mentioned as being instrumental in helping you become the scientist you are is Dr. Levy, who is an undergraduate bio lecturer at Georgia Tech, correct?
Yeah.
And Dr. Levy was instrumental in telling you about REUs. In fact, you did a few REUs during your time at Georgia Tech. The first one being in prokaryotic biology, correct?
Yep. Prokaryotic biology. And it was like down the road at UGA, University of Georgia. So in terms of the football rivalry, UGA was always Georgia Tech's football rival.
Yeah.
Georgia Tech was like a flea on UGA's back because they're the big football school. They're in the big conference in the Southeast. Georgia Tech is the more nerdy campus that we always were like picking this fight with UGA.
Yeah.
So it was always kind of interesting to me that like a good part of my undergraduate was at University of Georgia as well as Georgia Tech, just because there's just this within the state rivalry.
Yeah. But I think to say that you like had the full early career scientist experience in the state of Georgia, you're like, look, I've been at both institutions, not that they're the only institutions, of course, but they both help to shape who you are. And of course, we can't ignore the topic of that as being prokaryotic biology and the fact that you're a phytobacteriologist. I'm trying to connect the dots. It seemed like when I look at your CV and look at your first publication on Vibrio and look at your short time in Northrop Grumman, you were really moving towards medical microbiology. Is that fair to say, or clinical microbiology?
I think I was. I had a few different experiences. So at Georgia Tech is where I got to work on the Vibrio parahemolyticus work. So it's a mild gastrointestinal pathogen, the like weaker cousin of cholera. And that was more of a population genetics project that Tracy Hazen, my grad student mentor, was leading for her PhD. And then over a summer, I got to do the REU at University of Georgia, where I worked on a bacteria called Myxococcus xanthus, which is a really awesome bacteria. It is a multicellular bacteria. So they hunt other bacteria as like a wolf pack.
Yeah.
And then when they run out of food, they do a certain amount of signaling to then coordinate making fruiting bodies and then a small number of cells sporulate, and then that can allow them to potentially be transferred to a new decaying log or something.
Oh, that's wild. I didn't know nothing about that system, but it sounds incredible.
Yeah, and within that system, like the ecology of what these organisms do was really fascinating. But what that lab did was focused on some of the molecular genetics, exactly how some proteins interacted in order to secrete exopolysaccharides so that that was produced. And the two experiences helped me identify the level of biology that fascinated me, especially at the time. I remember having a conversation with one of my professors with, I've done these two research experiences and I really tools like PCR. I'm doing PCR in one lab and it just isn't really catching me. And I'm doing PCR in this other lab and I'm really excited. So I don't really understand.
Yeah.
And what he pointed out was the Vibrio perihemolyticus lab was focused more on population genetics kind of questions and epidemiology. And the other one was on more molecular genetic functions within the small scale. And I was like, oh, I didn't realize that like the tool could be used for different questions.
Yeah.
And that was like pretty late in my undergrad, and I'd had so many opportunities already. So it's just like humbling to be like, oh, the difference there.
Yeah. But that's a really good point because I think that if you ask a student, an undergrad about their experience or learning a certain tool or technique, they might say, "Oh, I didn't like that for that very reason." They were asking and answering questions that they didn't feel connected to. Or maybe the mentor themselves or the instructor wasn't inspiring them. It was just like, "Oh, this is the way we get the answer." But it wasn't like, what does that answer actually do for us? And how could it inform decisions 4 steps down the road? So sometimes it's like if a mentor or an instructor doesn't take the time to step back and take that 10,000-foot view, then you can't inspire your students to work harder and to maybe like to be hungry for that.
For sure. So the scale of biology that you're interested in is a question that I ask in a lot of my mentoring conversations with undergrads when they're trying to decide what they want to do. I talk about you've had your experience with us, but think about your classes and about the different topics that you've studied. Are you really drawn to the molecular interactions? Are you really drawn to cell, organismal, ecology, evolution? Like, where in this continuum is your happy place? And like,
Yeah.
how far are you willing to go up and down from that?
Okay, so you finished your degree in 2010, and then after that, is that when you worked as a technician at Northrop Grumman?
Yeah, it was a gap summer. And so I was a federal contractor through Northrop Grumman, which is a largely defense contractor, but they also have a small wing that staffs temporary positions at the CDC. So the Centers of Disease Control and Prevention in Atlanta. And I wound up spending a summer as a Sanger sequencing tech at the CDC.
And during this time, had you already applied to the University of Wisconsin to do your PhD?
Yeah, definitely. I'd already applied, interviewed, and accepted at the University of Wisconsin. So this was a nice experience that was able to fill the months in between.
Yeah. It's funny when I think of gap summer, I think of backpacking across Europe, not necessarily working, but maybe it's a little bit harder to justify a backpacking summer.
Yeah, I think that's probably true.
You go to the University of Wisconsin after that summer. So we're talking like the fall of 2010.
Yep, fall of 2010.
Do you go and do rotations or are you joining a specific lab?
Yeah, we do rotations through the department I joined. So I joined the umbrella microbiology program, MDTP, microbial doctoral training program. And that program does 3 rotations. It's funny looking at my application statement to the university, because in those you often identify some professors and research who interests you. And the 3 professors I happened to list in my application, I didn't even contact for rotations.
Yeah.
To some extent, I had no idea exactly what I wanted to do. I knew I was interested in molecular scale, but there's just so many potential niches that you can do in molecular bacteriology, I guess molecular microbiology. So I wound up rotating through 3 labs. Diana Down's lab, which focused on metabolism networks and using genetics. And with that rotation, I realized I loved my biochemistry classes. I absolutely loved the seminars that the people from the Downs lab were giving. It was too small for where I really found my passion as a researcher. And it was a little too detailed, like identifying a new enzyme that shunts some part of a metabolic network.
Yeah.
Was more chemistry than I actually wanted to do.
Okay. That makes sense. And was one of those rotations how you ended up in Caitlin's lab?
No. When I was selecting rotations, Wisconsin has a ton of microbiologists. There's a list of, I think, 90 professors who I could potentially select. And you can't read papers of 90 professors and make a really detailed decision. But they had this nice table that had a few keywords about professors' research. And when I looked at that list, I was biased by one of my great mentors. So my high school teacher, Sam Parks, she inspired my love of microbiology, but she wasn't particularly interested in plant sciences.
Yeah.
So when I had her for AP biology and there was too much curriculum to possibly fit into the whole year, she assigned the 3 or 5 chapters on plant science as take-home reading.
Okay.
I think the lesson I remember taking away was that plants are hard and boring to work on. And I was like, so plants are boring and hard? Bacteria are easy and fun. And I'm going to focus on bacteria and skip the eukaryotes.
Uh-huh.
And so when I looked down the list of 100 faculty at Wisconsin, anyone who had plant mentioned in their row, I just skipped straight past that. And so I wound up doing 3 rotations that were in very different fields. I wound up selecting a thesis lab that focused on protein trafficking in single-celled trypanosomes, the eukaryote, which was like exciting for me. I was like, oh, well, look at me being adventurous and trying something that's not a bacteria.
Yeah.
But I can always come back to bacteria, it is what I felt. About a year into my lab, just after I had made my own intellectual contribution of working with one of my classmates to apply his great bioinformatics expertise to make hidden Markov models, to identify more examples of my proteins in my little trypanosome genome. So I just made this contribution of expanding the scope of what my thesis would be. My major professor unfortunately announced that he was moving his lab. And I had this like pretty terrible, traumatic experience of having to decide, do I follow like my advisor and move my project to a new university, or do I stay where my partner was? So my boyfriend at the time, now husband.
Yeah.
And like I was like, I've got this cohort that like when I applied to the university, not necessarily to the labs. The people I thought I was interested in, I didn't even rotate.
Yeah.
So I wound up going as a second-year grad student, wound up going back to the list of research professors to find someone who maybe would be interesting and maybe take me as a lab move refugee.
And that's how you found Kate.
Yeah, that's how I found Caitlin. And it's like the whole experience of my first thesis lab, I consider an unofficial master's because I grew up a lot in that year, both in terms of understanding my scientific interests as well as how to try to structure my time in the unstructured research workspace. And as far as like research interests go, I recognize that bias that like I had just accepted, the bias being that plants are boring and hard, and so you could skip them. I'd had a great microbial symbiosis course by Heidi Goodrich Blair that talked about how microbes interact with all sorts of organisms, between microbes and animals for both beneficial and negative, microbes and plants for both beneficial and negative. So the rules are really similar across these different systems. And the molecular mechanisms in how these organisms interact are conserved across a lot of these systems.
So what you learn in one thing can be applied to another.
Exactly. And so I was like, oh, so that's cool. There's this whole field of plant science. We also had a graduate program structure where mid-stage PhD students would present their research several times throughout their thesis. And one of Cadel and Allen's students had presented this really cool talk about how this pathogen uses nitrate as an electron acceptor, and how this is relevant to how its infection cycle because it doesn't have a lot of oxygen after it's consumed a lot of that in the plant. And I remember really enjoying that seminar, and my takeaway after it was, oh, that's really cool that they were able to do such cool work in a plant system. I was recognizing my bias. And it was like 2 months after that great seminar that my professor announced that he'd be moving his lab. And I wound up prioritizing Cadel and Allen and the other professors in the plant pathology department for emails to see if they'd take me for a second-year rotation.
Thank you for all those details. And I think it's a good point to make that we could change our minds. We started this off by talking about how you became this accidental plant pathologist, but you also made the point that you stayed one because of the work, and that it's important, and that the community is welcoming. And it reminds me of this idea that people always want to know how you, like in general, how you met your true love. But they never ask, how did you stay together for so long? They only want your origin story. And your origin story of plant pathology is, I came into it like a lot of people came into it. I didn't know about it. Once I found out about it, great. But your staying in it was deliberate, and it was your choice. And I suspect that the strong mentorship you received has a lot to do with it.
Yeah, definitely. The honest story is, after making the lab switch, there was some psychological, emotional trauma to heal. It was during the switch that was my first period of depression, where it had this vision of how my next 5 to 6 years was going to go. And for nothing that was within a modicum of my control, that was ripped out from underneath me. And I had to make a switch. So I was there was a period of lack of security in terms of my career stage. And then a little bit of just being grumpy about the academic system, because the nature of it made me pretty sour on academia. But then luckily a PhD in the US is quite long. So I was able to heal from that and eventually start having fun again.
Yeah.
In research. So I was definitely motivated when I hit the new lab, but that's part of the healing process.
Sounds like you went through a boom-bust cycle.
Yeah, I definitely went through a boom-bust cycle. And then the lab environment that I joined was excellent in Caitlin's lab. She was a really supportive mentor. Yeah, and just, provided infinite opportunity.
Remind us again what you worked on there.
Yeah, so that's where I did meet, I guess, my true love, besides my husband, sorry, my second love, my research love, which is a bacteria called Ralstonia solanacearum. And Ralstonia is a great bacteria because it's one of the few bacterial plant pathogens that causes a charismatic, obvious disease for humans to recognize without understanding the impacts. So I remember in one of the seminars I gave to an audience of largely biochemists at Wisconsin, biochemistry focused PhD students, most of them focused on a few amino acids of a protein for their whole thesis. And in that seminar, I showed a time-lapse video of a tomato plant succumbing to bacterial wilt disease. And someone in the audience gasped and emoted with a plant, which was just amazing to me to see, because there's bias against plant science. We don't see them in our environments because they're very different organisms. And so we tend to a lot of, both in society and even as scientists, tend to not notice the fascinating biology until you are immersed in it to some extent.
Yeah, I think something we share in common is I worked on a verticillium wilt for my PhD. So I worked on vascular wilt disease, a fungal disease, but a vascular wilt disease nevertheless. And I recall I was doing some kind of windshield surveys for verticillium wilts on different tree hosts along the roadside. And I came into a neighborhood where there was a catalpa tree that was wilting in this guy's front yard. And it was a big catalpa tree. And I pulled up and knocked on his door and I said, "Excuse me, I'm a researcher from Penn State and I'm interested in sampling your tree so we can isolate it from this out." And he goes, "I have a tree in my front yard?" I was like, oh, yeah. I said, you have a big tree and it has a vascular wilt. And I took him outside. He didn't even notice there was an 80-foot tall catalpa tree, let alone a vascular wilt. So it was funny. But it reminded me of what you were saying, that like, if you are just oblivious.
Back to the lab.
Yeah, he's like, if it's dead, I said it could recover. Like, it's obviously has an acute wilt. But I said, I've seen this before where they'll just rebuild their crowns. He said, I don't care. Stop talking and just leave me alone. So I got my sample, and sure enough, it was verticillium wilt. But coming back to your point, it's really nice when you can easily demonstrate the negative, detrimental impacts that like a plant pathogen is having on a plant host.
Yeah, and one thing that, like, just with talking to different folks and trying to explain what I do in casual conversation, one of the, I don't know, my small amount of SciComm and outreach is I'll introduce myself as saying, I study a bacteria that kills plants and it causes the plant version of a heart attack. And so that connects it to something that everyone understands to some extent. For humans, that's a common problem that humans can face. We might know someone who's had a heart attack, hopefully recovered. And so that immediately gets people thinking. And then I say, but plants don't have a heart. This bacteria clogs up the vascular system that is necessary for the plants to live. And so the plant version of a heart attack is a little slower and it's a wilting disease. And so that usually it's kind of that hook to connect new information to something that they understand in their own experience.
Yeah. I know we don't have forever and a day to do this podcast. We're limited in time, but what aspects of Ralstonia did you work on during your PhD?
So it came back to biochemistry during my PhD, but more at the organismal and interaction scale than the metabolic network and identifying enzymes scale that didn't quite capture me.
Mm-hmm.
So my PhD work focused on several aspects of the metabolic interaction between Ralstonia and their tomato hosts. I studied two different pathways that the bacteria has to break down some chemical defenses that plants wield against pathogens. Plants are great chemists. They tend to use a lot of chemistry in their defense against herbivores and pathogens. And two of these aspects are phenolic molecules that Ralstonia is able to degrade. One being hydroxycinnamic acids, which are present in root oxidates and precursors to the polymer lignin. Another being salicylic acid, which is a key defense hormone for plants. And both of those are chemicals that Ralstonia has multi-gene pathways to break them down and turn them into food. So they're able to break down these defenses. It's probably not like the most useful food, but it's a little cherry on the
Yeah. So when I look at your publication record, you have several dozen peer-reviewed papers, and I found that all but two of them actually have Ralstonia in the title. So it's clear that Ralstonia has not been solved. It's still a problem. And besides tomato, what are the major plant hosts of Ralstonia? And you work particularly on a species that impacts tomatoes and possibly other hosts. But do Ralstonias as a genus, are they like problematic worldwide? Or are they more a problem here in the Americas? Tell me a little bit more about that.
Yeah, Tiffany is a global pathogen. There's a lot of diversity within Ralstonia. The locations where they tend to be in the world tend to be definitely tropics or warm temperate areas and regions that receive year-round precipitation. Where I'm at in California is not really impacted by Ralstonia, unlikely to become established here, especially with the direction that climate is going. But in the US, it's an issue in the Southeast, an issue in Hawaii, Guam, and Puerto Rico. And it definitely causes huge impacts in the global tropics, especially for smallholder farmers who, when they get an infestation, it's a soil-borne pathogen, so those are always really difficult to manage. It can persist for many years. And then the other aspect that's really especially hard for smallholder farmers is, unlike most bacterial plant pathogens, Ralstonia are generalists, and that's not at the genus species level where we say that Xanthomonas can infect a lot of hosts. But when that's said, it's usually any given strain of Xanthomonas has a fairly narrow host range,
Yeah.
and collectively they infect a lot.
Okay.
But Ralstonia are these like complex generalists. They definitely have patterns and limits to their host range, but it's extremely complex. So a farmer can potentially bring in a strain on potato tubers that they plant, and then it might have broader host range to tomato. Sometimes the potato strains and banana strains can be the same, but there can be host range that is across the monocot dicot divide. Yeah, so that ends up creating a lot of problems. When I started my lab, understanding host range of Ralstonia was something I realized was a huge gap in knowledge for the field. And especially on the more basic side of trying to understand the molecular rules for why certain strains infect different plant hosts, Ralstonia was skipped as a model organism because its host range patterns were too complex.
Okay.
And we still don't even have words to describe the patterns because it's just each strain can be so variable. It's genetically determined in their genomes, but we don't really understand the specific ways of that. So when the COVID-19 pandemic hit, which was 8 months into starting my lab at UC Davis, we all got sent home. And I had some undergrad mentees in the lab who were part of our major, the global disease biology major. And to graduate, they all do a research practicum. And the wet lab practicum that we had planned for them was clearly not going to happen before they graduated.
Yeah.
After we figured out how to get groceries at home, it was to figuring out how to work from home. In my lab, I had adapted a few new model strains of Ralstonia. And then I wanted to ask genetic questions that were ecologically relevant to them. So I wanted to know, what are the host range of these strains? Where are they located? What can we understand about their context? And from reading the literature, I was like, wow, there's really not a review that like summarizes all of these population survey papers. And there's so many of them. And it takes a long time to read it. It takes a long time to start putting it in my own Excel spreadsheet. And to flash forward to the pandemic and we have to work from home, I suggested to Kyle Chipman that he should take this Excel template I started that just had a lot of columns: host, location, strain name, paper, and some different information, and go through Google Scholar and download papers, grab the tables of strain data from those papers and put it into Excel. And it became a practicum that we've mentored maybe close to 10 students on. And at this point, we have information from over 300 papers, 10,000 strains. So we had this huge data set. And I'm someone who is willing to comb through Ralstonia data and do some sorting and some organizing that data to see patterns. But I realized that even most plant pathologists don't have the time or interest to go through this huge spreadsheet. So we were able to get APHIS funding to make a dashboard. Also inspired by COVID there, because we'd all checked the COVID-19 dashboards to see is incidence increasing, where is it? We probably all followed the next strain phylogenies to see these lineages emerging. And what we do is we're not doing next strain tracking, but we're just trying to consolidate the patterns that exist and are described so that we can get a bird's eye view. And so now we have this web-based dashboard that has interactive maps where you can like filter by different phylogenetic groups, and some like cool stacked bar charts that show for one of the phylogenetic groups, phenotype 1, what are the plants that those tend to infect. And there's some patterns of host range that I didn't understand from reading the literature. From reading the literature, tomato and potato are both major economic hosts that are impacted by Ralstonia. And then the papers will also say eggplant and pepper are also hosts, and these are all Solanaceae. And probably like many people, I assumed that if a strain infects tomato, it's probably infecting all the Solanaceae.
Sure.
But from looking at this incidence information across the globe, one of the major phylogenetic groups, phenotype 1, accounted for the vast majority of isolations on pepper and eggplant. But then if you look at like potato, it's way more balanced among the phylotypes.
Okay.
So it was just interesting to see this kind of force.
Yeah. And I want to talk about that kind of thing you said about not everybody wants to tackle the spreadsheet thing. I actually want to come back to that. But before that, something that really caught my eye and preparing for this conversation we're having is, so Ralstonia is named after a researcher. Is it Erica Ralston? And she did her PhD at Berkeley. You did a postdoc at Berkeley. And she was a professor at UC Davis. And you're a professor at UC Davis. You also said it's not likely a problem, or it's that Ralstonia is not really a problem in the greater Davis area. Yet, several of the most important researchers in this area have worked at UC Davis, and I think it speaks to UC Davis being a really important agricultural powerhouse. But I find that to be amusing that you went on to work on Ralstonia, following maybe in the footsteps of Erica, and having the same lineage in some ways. That obviously is something that you've thought about, right?
Yeah, definitely. There's a few things there. In addition to myself and Erica Ralston, but probably better known as Erica Barrett, in addition to us, there's also Ivan Budenhagen. And Ivan is one of the founders of the field of Ralstonia. He was a professor at UC Davis. His expertise really was on banana agriculture, especially banana pathogens, but also breeding. And he was just this global banana expert. In the 1960s, Budenhagen and Louis Sequeira were stationed with one of the large export banana plantations to solve a problem in South America of this new epidemic that turned out to be moco disease of banana, and that's caused by some of the strains of Ralstonia. And so they were spent years trying to understand this epidemic and discovered that unlike most Ralstonia that are soil-borne, there's weird aspects of the banana growth patterns and the ecosystem that they're in that was enabling insects to contact spread Ralstonia from male flowers of the different banana trees. So the male flowers would lose their petals and leave this oozy wound. Insects would find that full of sugar and bathe in it. If that happened, that oozy wound was full of xylem sap. If it happened at Ralstonia, they would move side to side. One more tie-in. Louis Sequeira is actually in, I guess I am actually in Louis Sequeira's direct lineage. After that time, he established a lab at University of Wisconsin-Madison. Towards the end of his career, a great postdoctoral scientist, Caitlin Allen, worked in his group. She wound up securing a faculty position joint between the plant pathology and the gender studies department. Founding her lab, she inherited this UW collection of Ralstonia. I did a PhD with Caitlin, and when I started my lab, I recognized the gold mine that was that historic strain collection in her lab of there's just so much to explore genomically of those strains. And so I went back and tried to replicate as 300 isolates from around the world from her lab. So there is that lineage connection. And then I joined an institution where Ivan Budenhagen was at. So then I had these two founders of the field of Ralstonia, and I've been there.
That's great. Thank you. We were talking about this kind of overwhelming desire to compile data. I too suffer from that. And for me, it's actually a quirk of having ADHD. Do you think that some of your ambitions are driven by your ADHD?
Absolutely. It's a phenotype that has its strengths and its kryptonite. Sometimes it's on the superpower side, sometimes it's on the kryptonite side. But that's everyone has their phenotype. And so some of the success is figuring out your unique special thing and figuring out how that matches with an important problem and trying to follow that and then make sure that you do the other things in your life and job that are also essential.
Damn you, administrative tasks.
For sure. So when you described the deep dive you took during your PhD, it reminds me of, so my third paper for my PhD was a metabolomic comparison of xylem sap of healthy tomato plants and tomato plants that were succumbing to Ralstonia infection. And I did this big experiment, really careful, really stressed, but lots of record keeping. So I would have my good samples, send it off, and it took several months to get the data back. And I got this data back and I was excited, a spreadsheet. And sorting it, it's clearly there's this one compound, putrescine, that was so much more present in the infected plants than not. And I was I took a biochemistry class and I like metabolism more than most biologists. I'd never heard of putrescine. What the heck is this?
Yeah.
So I think I got that data set on a Friday. And over the course of the weekend, it was just me in my pajamas and a sweatshirt because it was winter in Wisconsin. So I was sitting in my old rental house, just downloading hundreds of papers on putrescine to try to figure out what is going on. So my questions were, what is this? And if it's being more enriched during interactions between Ralstonia and the tomato, is it likely coming from the bacteria? Is it coming from the plant? Is it a bacteria attack? Is it a plant defense? Like, why is this
Yeah.
becoming enriched? And the patterns were extremely complex because it does turn out that I call putrescine a primary metabolite that no one talks about because it's present in all life, but it's not food and it's not a building block of like DNA or protein, so it gets ignored.
Yeah.
But it has like major tie-ins to reactive oxygen species metabolism across life, which at a mechanistic level makes it a hairy mess to figure out. Because like ROS can be a defense signal, but then it can also be like a too much reactive oxygen species can be useful, but it can also be harmful and they're really labile. So it's hard to really like design experiments to get the right time point to test anything.
Yeah. Yeah.
So I wound up not being able to go too far in that. So I was like, oh, this thing that's connected to everything, polyamines, putrescine and polyamines, is connected to ROS, the other thing that's connected to everything.
I don't think you're giving yourself enough credit. I think you did show that it was associated with symptom development, right? Like enhanced wilt production or enhanced wilt was associated with this increase in putrescine, correct?
Yeah, we showed that adding extra putrescine to plants would increase wilt production, and we made a somewhat of a circumstantial evidence because of limitations of biology. We made a robust but circumstantial case that the putrescine that was being produced during the interaction was coming from the pathogen. I think that there's a recent abstract of a paper I haven't fully read yet from Caroline Baroque's lab that's been able to tackle that question from a different angle to say, is the putrescine coming from the bacteria advancing wilt or not? And the answer might be maybe not. I don't know.
Yeah.
There's only so much that we can conclude. So she might be able to solve the gaps a little bit better.
Okay. In 2025, you were promoted and received tenure. You're now an associate professor. Congrats on that. And in 2024, you also were the recipient of a NSF CAREER Award, which is a big deal. And certainly, I want to spend some time talking about that and its tie into some of this work you're doing with the cohort-based undergraduate research experience, which I suppose is a big part of the mentoring side of your career grant. So will you talk a little bit more about your career award and this CURE project you're working on?
Yeah, cohort-based undergraduate research experiences are a great way to expand access to research training. The experiences I had were phenomenal, but what I realized is that it's not scalable because it was very intense hands-on training of one grad student worked with me and maybe a second undergraduate, but that's not that scalable. And then also looking at my peers, not everyone wants to do research, and that's okay. But it's important for many people to get the opportunity to say, is this the kind of work and problem that you want to pursue? And then I noticed that some of my peers would get those opportunities and learn a really useful lesson, which is this is absolutely not the kind of work I want to do in my career. And I'm much
Yeah.
further suited to maybe clinical testing or something that's a little bit more routine and extremely useful. But researchers need to be driving forward new knowledge, and that's hard and exciting. So undergrad research experiences provide more opportunity. And I learned about them because of the C-phages program, which is started at Pitt, I think, by Graham Hatfield. And that focuses on students going out and isolating bacteriophages that infect different bacteria, sometimes acne-causing bacteria or something that's a little bit fun. So they go through the isolation, so some of the microbial ecology, and that's like a semester. And then they would do genome sequencing and analysis of their phage genome in a second semester. So I heard about that and I was like, oh, this is phenomenal. And I was like, it's doing two things. It's giving people a lot of access. And also when I compared that to like what we were told was a good PhD worthy project in my PhD program, I was like, they're also doing work that I've been told is below PhD quality or scope. It's below PhD scope
Yeah.
because it's repetitive. It's what molecular biologists can sometimes derogatorily call stamp collecting. Going out and describing diversity is sometimes not always it's not always respected in science, and it can definitely be hard to fund because to some extent it's like, why go study how diverse and why go study that population? Why go spend a lot of time there versus any of the other understudied systems? So sometimes there can be difficulties in studying ecological diversity So what I saw from that cure and then hearing about other cures at like a conference was it seemed like a lot of these cure projects, you're also working with noobs, right? They're beginner scientists. They don't know how to hold a pipette probably. There's so much that they have to learn in order to get even their own understanding of what they're doing, let alone real research and publishable data. Parallel methods are really important. And so that kind of comes into if you're going to do parallel methods as you train these students on science, what is useful that can be done in parallel? Studying some diversity questions, like collecting stamps. And there still can be hypothesis driven work. There can be exploration. And during my PhD, I was really excited about trying to make a cure if I became a faculty. But at that time, I was doing like primarily genetic engineering. And I was like, what I would train my undergrad mentees on was cloning and knocking out genes to make some sort of mutant. And so they learn PCR and all these things. And I was like, okay, so this could be very scalable where we could have students by the end of the semester have a mutant of a bacteria. And then I was like looking at my PhD work and I was like, and then it takes a lot of effort to make that into new knowledge. Like you have to then test it and go figure out and go unexpected things. And I was like, I don't see how that's going to become a paper. Like you can't just say, here's a mutant. So that didn't seem like useful parallel knowledge to make at the time. And so I was always trying to think about what would be useful to a research program, but scalable as a research question. And part of the time of sitting at home during the COVID-19 pandemic and interacting on social media with scientists to figure out what are people doing, I realized that the cost of whole genome sequencing had come down a ton. And there were these companies that could provide a genome sequence within a week as well. So that actually was now really feasible for a semester or quarter timeline. Students can get data back, and the costs are low enough that we can potentially sequence a bunch.
Sure.
So my cure takes advantage of the great resources of strain collections of Ralstonia, that some of which were housed at University of Wisconsin, and I was able to inherit via Louis Sequiera and Caitlin Allen. Also collaborations with Sally Miller and some other plant pathologists around the country. And each winter quarter, we recruit about 15 students to each sequence the genome of a new Ralstonia strain. And so we send it off for whole genome sequencing. It's a bunch of big data. We use this great platform called K-Base, which doesn't require them to have the technical prowess of coding. It's a graphical user interface that can do all of the big computation on a remote server, but it also teaches them lab notebook skills because it's a Jupyter notebook and they go cell by cell. We basically had just this one big manual, reserve a time and let them go at it. The first week, students were always really like apprehensive of when am I supposed to get this done by? Yeah. And you can just see the high performing anxiety that's also tailored well to the classroom education. There's going to be a test and you have to get certain things done by deadlines. And in the cure, we get to train them on big projects, professionalism, dealing with an unstructured task. And when they ask, like, when am I supposed to get this done by? I say, it depends. You could speed through it, just click all the buttons and get to the endpoint really fast. Or you could really look at your data. You could take good notes. You could read more of the documentation, read some papers, and really drive into your interests. So I really can't tell you, but we can keep talking as a group about where people are. And some people might get ahead and they can potentially provide advice back to each other.
In reading over your materials, the few things that jumped out to me that I found interesting and I really respect is that one is that, yes, you're integrating undergraduate research into a program and you're having them, but in your program, it's taking community college students, transfers. So it's a very inclusive environment because those students very likely didn't have any previous experience or limited experience because of just the lack of resources maybe at the community college level. And the other thing I was reading about is like this idea of, in a way, it's like a flipped research experience, right? You dive into the data before mastery, as you say. And I think that's really great because so often when you're trying to get a new undergrad interested in something, you're like, you have to do all this boring work. And 2 semesters from now, when we publish the paper, we'll tell you what it meant. But this way, you're showing them some of the exciting results before they even like master the subject. So I think that flipped aspect of it is great.
Yeah, I think there's two aspects. Like understanding why they're doing stuff, that always takes time, even with this aspect. But my experience, and especially thinking about, I think a lot of the molecular biology focused students who get into it, a lot of the excitement is learning how to move liquids to do a PCR. And so there's just so much about liquid moving and how you have to do that and don't leave the enzyme out. And there's all this like hands-on information that you have to learn in order to get that successful result at the end. Sometimes students are actually more interested in the technique than the knowledge. That's okay, because there's careers for both directions. But with the flipped research experience, day one, they click a button and they get quality control results on their reads. And it's 30 graphs that all have different axes and they're labeled. And they're not necessarily the best formatted graphs, but they're functional thanks to the tool. And they just have to like look at data. By the end of the quarter, they still get an appreciation of what some of the big questions are, but it helps them understand that, especially more and more, a lot of research is going in the data analysis direction because data analysis is so scalable. So there's always that struggle of some people are really great wet lab or field people, but man, getting them to analyze their data or getting themselves to analyze their data is just a struggle.
Yeah.
But like maybe in industry, that's fine, right? If you want to be the experimental design person or the experiment running person, you don't always have to be the data analysis kind of person. And so sometimes these roles are more compartmentalized in other fields. In research, you have all of them.
Yeah.
And so they get experience with that first.
It's been really exciting hearing about your work, and congrats again on tenure and the Career Award. I appreciate you and what you're doing for our field. It was great talking to you.
Great talking to you too.
We've just heard from Dr. Tiffany Lowe-Power, Associate Professor of Plant Pathology at the University of California, Davis. As with all our episodes, be sure to check out the show notes immediately following the episode description for links to Dr. Lowe-Power's research program. I'm Matt Kasson, host of Plantopia. Thanks for listening.